Your voice. Good evening, everybody. Welcome to tonight's Leeds Fill and Lit talk, which is going to be given by Professor Nick Tosca. My name is Eric Blair. I'm president of Leeds Fill and Lit, and I'd like to give you a very warm welcome to tonight's talk. Both our own members, and if you're not a member or indeed a recent member, you're all very welcome to this talk. At the end, there will be time for questions, but you can, during the talk, write questions or comments in the chat, and Rachel and I will read them out, or you might want to ask your own question at the end. So without further ado, I'd like to pass over to Rachel, who's going to introduce Nick.
Thanks, Eric. So Professor Nick Tosca from the Department of Earth Sciences at the University of Cambridge is part of a team of scientists at the core of the Mars 2020 Perseverance mission, and the only scientist from the UK to be selected by NASA for a role in this work. On hearing that he'd be one of the first scientists to analyse Martian rock samples when they're brought back to Earth, he reacted with, the fact that we may actually see these samples and study them in my own lifetime is just mind-blowing. And when I told my son about it, I think we pretty much had the same reaction. I kind of felt like a 5-year-old in how excited I was what discoveries this mission might bring. And just that raw curiosity and interest in other worlds that most of us had when we were that age. So his research group focuses on understanding the co-evolution of life and environments through Earth's early history, using laboratory experiments and theoretical modelling to help them to understand and work out connections between environment and biology. And Nick will explain, I hope, how understanding the early evolution of sedimentary environments on Mars could indicate whether physical and chemical conditions on that planet were ever supportive of microbial life as it's currently understood on Earth. He did his undergraduate and postgraduate and postdoctoral work in the States with postdoctoral studies at Harvard. And from there, he came to Cambridge as a research fellow, then had a spell in St Andrews, and then he was in Oxford, and then last year arrived in Cambridge, and that's where he's speaking from tonight. So I'll just pin your video and hand over to you, Nick. Thank you very much.
Okay. Thank you very much for that kind introduction, Rachel. It's a real pleasure to be here with you this evening and to share some of the exciting science that really forms the basis for the Mars 2020 Perseverance Rover mission. Before I begin, can I just check that everyone can see my first slide, which is the title slide here? Yes. Seeing nodding thumbs up. Okay, excellent. Yes, so this talk is put together in 3 parts. The first part, I'm going to discuss the scientific motivation behind the mission. And in the second part, I'm going to introduce you to the capabilities of the rover and the type of data that we're collecting. And in the third part, I'm just going to speak briefly about the results that we've collected so far and how they might relate to the very beginning of the initial motivating science behind the mission. I'll say right away that being involved in this mission has been an absolute joy. And I speak the truth when I say that I'm experiencing this mission through the eyes of my 5-year-old son. And I think it's best to do so because when we're confronted with unusual data, I think it's important that we all keep an open mind and don't bring any preconceived hypotheses to the table, because as we know, geology is complicated, especially when looking at geology on another planet and geology that was formed and modified about 4 billion years ago. So some of you may be familiar with the Mars 2020 Perseverance Rover mission because engineers at NASA decided it would be a good idea to install a series of cameras to film the entry of the rover, or the space vehicle and the rover, through the Mars atmosphere and the successful landing of the rover. And the rover is about the size of a Mini Cooper for scale. So it is an enormous and complicated piece of instrumentation. And the fact that NASA managed to land this safely on the surface of Mars is nothing short of a miracle. But it's even more of a miracle if you understand exactly how that landing took place. And so if you haven't seen the footage from that landing, I'm going to show you a short clip now, complete with sound, so you can experience the last thrilling moments as the space vehicle hurtled through the Martian atmosphere and the rover touched down safely on the surface of Mars. So I'll start with that, and then I'll continue on with the talk.
Here in Safety Bravo.
We have completed our terrain relative navigation. Current speed is about 30 meters per second, altitude of about 300 meters off the surface of Mars.
So this is actual footage from the series of cameras that installed on the rover.
Accordion, which means we are conducting the sky crane, about to conduct the sky crane maneuver. Sky crane maneuver has started, about 20 meters off the surface.
We're getting signals from MRO. Tango Delta.
Touchdown confirmed. Perseverance safely on the surface of Mars, ready to begin seeking the signs of past life.
So that happened on the 18th of February, 2021, and received a huge amount of international press attention. But what's not as well known about this mission, aside from the fact that the Perseverance rover is looking for signs of ancient life on the Martian surface, is that the rover is selecting the samples that will be returned to Earth within about 10 years. And I'll explain exactly the nature of the samples and how those samples are being collected. But basically what it's doing is selecting the samples that the science team believes stand the best chance of preserving evidence of ancient life within the rock record. The big question here is why? Why do this? Or indeed, why bother? One could easily make the case that looking for signs of ancient life in the Martian surface is the longest of long shots. And part of me feels that that's the case, but there's much more to be gained scientifically by going through and carrying on with this experiment. And part of the reason relates to understanding our own origins. And in the last 10 or 20 years or so, I think it's safe to say that chemists and biologists and earth scientists interested in studying the origins of life have experienced a revolution in understanding the processes that might have progressed simple sort of multi-molecular chemical systems to systems that are capable of replication and perhaps even metabolism. Now, this is showing a video animation of vesicles formed from lipid molecules or fatty acid molecules, and the ways that those vesicles interact with each other and can actually form primitive compartments or membranes. And so this is a video that was put together by Jack Szostak's lab at Harvard. Now, aside from understanding the origins of compartments and understanding the importance of compartments to early cells, one of the big revolutions in understanding the origins of life comes from prebiotic chemistry. So understanding how the building blocks of life were synthesized from raw materials in Earth environments. Now, this incredibly complicated chemical synthesis scheme summarizes an enormous leap in our understanding of how key building blocks for life could be synthesized from one multi-component system. And so this paper was published in 2015. It came out of a lab that's based in Cambridge, the Laboratory for Molecular Biology. And these authors showed that the synthesis of ribonucleotides, so the building blocks to RNA, precursors to lipids, the molecules that are required to form cell membranes, and amino acids, the building blocks of proteins, could all be synthesized from simple raw materials all together in one rather complicated reaction network. And this has been an absolute breakthrough in our understanding of how one could progress from chemistry to something close to biology, really understanding how the building blocks were formed. I've highlighted on this scheme some of the important raw materials. In blue, HCN, or hydrogen cyanide. in orange, inorganic phosphate, both of which are supplied from the natural environment. Those two components, in addition to UV light, which is shown in yellow, are crucial to this entire scheme. And on the basis of those environmental requirements, these authors later suggested an environment or a sequence of events that could have taken in one rather complex environment that could have initiated this chemistry and modified it such that the building blocks could be synthesized in one place. And that's what this schematic shows here. So these authors envisioned that you would start off with a lake, which was open to the atmosphere and receiving ultraviolet rays from the sun. And within that lake, there was cyanide and phosphate. Phosphate is thought to be supplied principally from the surrounding rocks, and hydrogen cyanide potentially from the atmosphere, although that's a different conversation. And waters eventually evaporated, going through arid climate cycles to make salt minerals. Now, if those salt minerals were thermally modified, so if they were heated by impactors or metamorphic events or neighboring volcanic events, then some of those salt minerals could be transformed into incredibly useful precursor products that could aid this synthesis scheme. And so then if those materials were re-wetted, as is often the case with lakes on the Earth, they experience environmental cycling, then those components could be delivered into solution and the chemistry could effectively take off. Now, this is a really thought-provoking paper because it, for one of the first times, it suggested a specific environment that was compatible with what we know about organic synthesis of key biomolecules for life. But the big question is, what sorts of environments were present on the early Earth around the time that we think life originated? And could they have facilitated chemical pathways like the ones that I'm referring to here? Now, this is just a rough schematic of the entirety of Earth's history, excuse me, spanning the Hadean, Archean, Proterozoic, and Phanerozoic eons. And so to gain some information about environments that were present at the origin of life, we have to reach as far back as possible to the earliest realms of environments that may be recorded in sedimentary rocks. There's a problem, though, in doing that, is that we barely have any sedimentary rocks that survive from that time period on the Earth. Now, this is a schematic of the total number of sedimentary rock packages that exists on North America as a function of their age. And you can see that the most recent sedimentary sequences are relatively abundant in North America, but there's an abrupt transition and the older ones start to fade away and eventually disappear. So we have very limited observational evidence that informs us of what the Earth was like at the time that life was originating. And that's a huge problem. That's a function of geology mainly, but it's a problem that's unique to our own planet. And this should give you a clue as to what the problem is, as to why we have so few sedimentary rocks that are left from that period. The problem on our planet is plate tectonics. Plate tectonics is a really efficient mechanism for shunting heat from the Earth's interior to the Earth's crust. But in the process of doing so, significant proportions of the Earth's crust, including ancient sedimentary rocks, get destroyed in that process. And geologists discovered a few decades ago that much of the ocean floor is actually rather young in a geologic context. And the oldest rocks have been destroyed, or the ones that have survived have been intensely heated. They've experienced very high pressures, and their capacity to inform us about ancient environments has more or less been obliterated. And so that's been the real challenge for geologists to really weigh in on the origins of life problem, is that we can't really say what the Earth was like at that time because we have so few rocks that actually preserve those environments. But this is where Mars comes in. Mars is different because it preserves a record of ancient environments that we think exists nowhere else in the solar system. And what I have on this slide is a schematic of the surface of Mars, color-coded as a function of geologic age. And all of the brown rocks are on the order of 3.5 to 4.2 billion years old. So they overlap with the period in time that we think life was originating on the Earth. And these rocks are present at the very near surface, and they cover an enormous portion of the Martian surface. So of course, they're accessible to us if we can only study them. And in fact, we have been studying them with orbiting missions that have employed high resolution photography and spectroscopic methods that can give us a sense of what minerals are present in these rocks. But more importantly, we've been studying these with landed missions, rovers, which can go and do the things that any geologist with boots and hammer would like to do in order to reconstruct the environments that, that modify the local geology. And so just to give you a sense, many of these very ancient rocks are in fact sedimentary rocks. They're the right type of rock. They're the right type of rock that archives physical and chemical information about the environments from which they formed. And so these are some relatively high resolution orbital images just to show you that exquisite layering pattern that can be observed with orbiters that testify to the rhythmic nature in which sediments were deposited in various environments in the ancient Martian surface. So this is a summary of some of the major events that occurred early in Mars's history, as is sort of constructed from broad brush geological studies. And on this figure, I have a comparison of Earth's geological timeline to Mars's geological timeline. So the two planets formed at roughly the same time in the earliest history of our solar system. But the major periods of geologic time are divided up differently, labeled differently in the two planets. But what you can see here, even if you don't really care to read the specific observations or processes that are reflected in this diagram, is that all of the action happened early on Mars. And then things quieted down and it started to become a cold, dry, barren place. And so early in Mars's history, there was vigorous volcanism in the form of the Tharsis Formation, for example, which is one of the largest collection of volcanic rocks in the solar system. There was an enormous flux of impacting meteorites to the surface, which shaped and modified the surface of Mars and probably had implications for Mars's climate. And then there's geological and geomorphological evidence for activity in the form of liquid water, valley networks and outflow channels that speak to episodic flood events that have been dated with cratering statistics that give us a sense that liquid water was actually present frequently on the early surface of Mars, and left its mark in the form of minerals that actually require water to form. And so this last panel on the bottom just gives a sense for the major mineral groups that have been identified in some of these layered units. Phyllosilicates, or more commonly called clay minerals, the type of minerals that we find in our backyard that make up quite a sizable portion of our soil. And then salt minerals, which are also called evaporites when they occur in sediments and rocks. So certainly early Mars was a busy place and it involved liquid water. And so the natural question is, was it suitable for life? And so one of the major motivating factors behind this mission and behind many of the previous Mars exploration missions is that we want to understand the processes that made the Earth and potentially other planets suitable for life. And we want to ask the question, did life ever emerge on early Mars? And that's an important question worth asking, because if we come up with evidence that life did emerge on early Mars, we would like to know how. But if we conclude that life never emerged on Mars, we would like to know why. Because from an environmental perspective and a planetary evolution perspective, the early histories of Mars and Earth are so similar. We would like to know what was different about the two and what the key factors were that really made the difference in terms of whether Mars could have been inhabited or not. So this question, I think, is very much worth asking. So I guess it's important to be precise in our definitions here. And so a natural question is, what do we mean when we say life? When we say life, most of us think of humans and plants and animals, the macroscopic life that shapes and populates our world. But when we talk about life, we are really referring to microbial life. Another way of categorizing microbial life is to refer to life as a prokaryotic life. And there's a major difference in the biological sciences between prokaryotic life and eukaryotic life. So prokaryotic life, to which microbes, bacteria, and archaea belong, are composed of relatively simple cells that have functioning membranes that allow metabolic processes to take place, and a collection of DNA, the information carrying molecules, and some other functional components. But that's about it. And actually, as a biologist friend of mine says, you can think of prokaryotic cells as basically a bag of DNA and not much else. Whereas eukaryotic cells, the category of life that we and plants and animals belong to, are the end result of billions of years of evolution. The cells are actually not, on average, the same size as prokaryotic cells. They're much larger. But what's more important than their size differences are the fact that eukaryotic cells contain specialized molecular machines that do specific jobs that really provide the platform for complex life on our planet. And most evolutionary biologists feel that the evolution of the eukaryotic cell is something very different and a very low probability event relative to the generation of the prokaryotic cell or microbes. And in fact, microbes dominated much of early Earth's environments, and really eukaryotic life didn't really become an environmental factor until much later in our own geological history. So all of this provides good reason for really expecting that microbial life is the type of life that might be present elsewhere, at least on Mars, the case we're concerned with now. So I want to talk a little bit about Jezreel Crater, the Perseverance Rover landing site. And this is a digital elevation map that's going to zoom in on the edge of Jezreel Crater. And you can actually see the arcing mountains in front of us, which represent the rim of the crater. And the circle represents the landing ellipse to which the rover landed. But you can also see this fan-shaped delta deposit, which is preserved inside this crater. Now, this crater was formed billions of years ago, about 4 billion years ago. And after the impactor formed the crater itself, the climate changed and water percolated through this crater and deposited a rather thick delta succession within the crater. And we can tell that because we can see outlined in blue the inlet channel that was carved, and in green, the actual delta deposit itself. And so now we're going to fly over the delta deposit with the strategic traverse of the rover outlined in white. And you can get a sense for the extent of the delta deposit. Now, the plan is to examine the delta deposit and sort of traverse around what are bound to be a rather challenging engineering obstacles, but eventually to get closer to the shores of the ancient lake, which are probably close in this region near the crater rim, and then eventually to ascend the crater rim mountains themselves in order to investigate ancient environments that might have been formed in the aftermath of the Jezreel impact. And so that really affords us the opportunity to investigate a whole series of different environments that may have once been present in the relatively long history of Jezreel Crater. So this final image is a color-coded image that illustrates some of the major mineral types that are present in the deposits in Jezreel Crater. Okay, so I'm going to spend just a little bit of time talking about what the rover can actually do, some of the instruments that it has aboard. As I mentioned before, the rover is about the size of a Mini Cooper, and it has 23 total cameras, and those cameras serve a variety of purposes. They're engineering cameras, relatively high resolution cameras capable of zoom for scientific purposes. And then, of course, the cameras that filmed the landing from various angles, the entry, descent, and landing cameras. But the rover has most of its scientific instruments located aboard the mast, or essentially the head and eyes that look like it's looking at you in this image. And then there's an arm, a retractable arm that is capable of collecting high resolution chemical and mineralogical data of rock targets. And there are two instruments on the end of that arm that really provide us with exceptional information as to what the rocks are made of and how they might record ancient environments. So this is just a closer look at some of the images or some of the instruments that are positioned at the mast. So the camera that looks like a pair of 3D glasses is the Mastcam Z camera, a high resolution, high zoom camera, a set of stereo cameras. And then the single lens up top is a camera called Supercam, which is actually a camera that operates through laser-induced breakdown spectroscopy. So that camera fires a laser at targets up to about 7 meters away and can measure the spectroscopic response as those elements and the targets are excited. It can actually determine the chemistry of targets that are not so easy to reach. So that's really an extraordinary capability. So here's a picture of the arm in action, just to give you a sense of the relative size of the rover and also the motion the rover is capable of. But another thing that the arm can do is it can scoop and drill and study rock samples. And so this is another important capability that the rover has that I'll talk about in just a moment. But first, a word about the two instruments that are positioned at the end of the arm. The first instrument is called Sherlock, which is a Raman spectrometer, and that's a type of vibrational spectroscopy that provides information about mineral and organic structures. And on the other end is an instrument that I'm very closely involved with, the Pixel instrument, which is an X-ray fluorescence spectrometer. And that instrument allows us to take chemical maps of targets that are about the size of a postage stamp. And that's hugely important because it helps us understand how minerals are related to each other in space and time. And it really provides exquisite information and allows geologists to uncover environmental history recorded in ancient rocks. And last but not least, as I alluded to earlier in the talk, the rover carries a caching system, or a system to drill cores about the size of pencil leads, or pencils themselves rather, store them in sample tubes, and to leave the samples on the surface for 2 subsequent missions to find, collect, and to return to Earth. Those missions are in production and they have firm launch dates. And in fact, they have to be launched by a certain time in order to take advantage of the relative positions of Earth and Mars in the next few years. So a related question here is, what is it that we're actually looking for? And can the rover detect traces of life given the instrumentation that it's equipped with? I think the best way to answer this question is that the science team is using this instrumentation to look for something that we often refer to as biosignatures. And the word biosignature is a relatively simple one, and I define it here as any object, substance, or pattern that only biological processes can create. So an example would be organic molecules that we know are only created by certain biological processes. And in fact, those organic molecules, or many organic molecules, end up getting preserved in sedimentary rocks for billions of years. Actually, the lipids that make our cell membranes and serve as the compartments of early cells are actually quite robust, and they can last a few billion years, or at least that's our experience with sedimentary geology on Earth. So oftentimes we refer to those molecules as molecular fossils. In any case, that's an example of a type of biosignature, but biosignatures can also be morphological as well. And so this is a great example of a morphological biosignature. On the left-hand side are pictures of a type of accumulation that is familiar to geologists and perhaps other scientists as well, as stromatolites. And stromatolites are produced when colonies of microbes form microbial mats together that populate the floors of lakes, so usually saline lakes in the modern Earth, but they covered large portions of the ocean in the past. And the interaction between sediments and minerals and that sticky microbial material that really exhibits a very complex set of chemical processes ends up leaving its mark in ancient rocks in finely layered and distinctive morphological patterns. And so this is what the instruments Sherlock and Pixel would see if it ended up analyzing something like a stromatolite. So just to give a sense of the type of morphological biosignature or something that we think could be very probably formed by microbial activity. Okay, so for the last part of the talk, and I'm just going to spend a few slides on what we've done and what we found so far and where we plan on going. So the first thing we did after we landed, aside from all the engineering and safety checkouts, was that the science team decided that we needed to explore the local geology. And actually there exists a fantastically detailed geological map of Jezreel Crater that was produced for the United States Geological Survey. And this is a portion of that map here. And you can see the black dot is the landing site. We landed in the crater floor, some ways away from the delta deposits, which are illustrated in darker blue. But we landed right near the contact between two different geological units, that pink unit and a sort of a salmon colored unit labeled NLE. And so this is a closer view of this salmon colored unit. And this is a photograph taken from orbit that shows the traverse that we're going to take once we're finished working in the area around us. So we're actually very close to this last point on the yellow traverse called Seta South. But we've traversed to that location from the Octavia E. Butler landing site. We're actually going to go back around the way we came and arc around this unit and eventually make our way to the delta. But in the process, we decided to investigate these two geological units that the rover actually landed very close to. This sort of bulging salmon color unit that I referred to in the map before, and the surrounding crater floor unit that lies to the east of this map here. So the big question from the science team is, what kind of rocks were we driving on? Were they volcanic rocks? Were they sedimentary rocks? I mean, this is a very basic question that any first year undergraduate student should know. But of course, no one within the science team was in a position to say for sure. And we weren't until we ended up getting our first detailed chemical and mineralogical data from the rocks. So on the left-hand side is an image of an abrasion patch that we drilled into the surface of the rock to allow us to study the fresh surface, so not the surface that's been subject to weathering and alteration for billions of years. And after studying that abraded patch, the protocol was to sample this rock, so drill a core through the rock and select a core under the understanding that the abraded patch was close enough proximity to the core that it would give us some information about what the core was probably made of. And you may have heard, because this ended up making some press, that once we took the image of the sample tube, it turns out that the first coring attempt had failed. This is an image of the sample tube that shows that the sample tube was actually empty after attempting to collect the sample. This, of course, sparked some great activity within NASA, especially the engineers who treated this as an emergency scenario and worked all various possibilities. But of course, a lot of the geologists on the team figured that the rock was probably too incompetent to withstand drilling. And that's exactly what happened. So this is an image of the core itself. And it turned out that the core was completely obliterated, ground into fine powder during the drilling process. So this is actually a nighttime image showing that the mound of powder left behind that coring attempt. So the question is, what is this stuff? Why is it crumbly and falling apart? And what are the minerals in it, and how did it form? And so this is an image showing all the different areas that we worked in the abraded patch to try to understand what it was made of, using all the different instrumentation, the Supercam instrument, the Pixel and Sherlock instrument. And I'll show you some of the first Pixel results taken from this rock. These are chemical maps that give us a detailed sense of the minerals that are present in this rock and what they have to say about the environment. So in this first map, this is a map of iron, aluminum, and chlorine. Iron is in red, aluminum is in green, and chlorine is in blue. And you can actually see that there are some areas where the iron and chlorine are mixing. Those would be the purple pixels, mixing between purple or red and blue. And this is a map of the same area, except this is a sodium, sulfur, chlorine map. You can actually see that the sodium and chlorine are now mixed together quite intimately, and there's a lot of sulfur within this rock as well. And so all of this suggests to us that there are salt minerals present here, so calcium sulfate salts, halite, which is sodium chloride, in addition to rock-forming minerals, so minerals like clinopyroxene, plagioclase, iron, titanium oxides. This is the stuff that volcanic rocks are made of. And there's this other very puzzling material, this material that's incredibly enriched in iron and silicon. It actually turns out that those minerals, just to show a little bit of the chemistry itself here, those minerals lie in a region of compositional space that are occupied by serpentine minerals. And iron-rich serpentine minerals are incredibly valuable to us because they typically form under relatively high temperature water-rich conditions. And so this is a schematic of how hydrogen and silica in solution actually combine under certain circumstances. So in this case, at 200 degrees C and relatively high pressure, to make a variety of the minerals that we've observed in these rocks. And so that this fits very nicely with our understanding of the chemistry required to generate these minerals. And basically, this concludes to us that volcanic rocks were emplaced into the crater floor, but they were heavily altered by hot water that generated iron serpentine minerals. This is a process that we refer to as serpentinization, and it often generates hydrogen as a byproduct. In addition, the waters that percolated through these rocks left behind salt minerals in exactly the places where the serpentine minerals formed. So all of this together broke apart the rock and heavily altered it, but it also means that the rocks themselves record a very complex history of water, probably involving multiple episodes. So in the end, we did end up with a successful paired sampling. So we sampled 2 cores from one rock of similar properties that I described before, except this rock was much more coherent than the first one I described. But I want to go back to the very beginning of the talk here when I was discussing environments that could be relevant for generating the building blocks for life. I talked a lot about the formation of lakes and salt minerals that were modified by thermal processes in order to generate the type of chemical building blocks that would be required for life. And as far as we can tell, this sequence of events is actually probable and potentially compatible with the data that we've already collected from the Mars Perseverance Rover. And that's kind of an exciting prospect. I mean, there's a lot of work we have to do to really understand the environments that deposited these rocks. But among the working hypotheses are situations like this, where a lake that was going through cyclic wetting and drying processes might have been influenced by the emplacement of volcanic rocks. And those processes could actually have great significance for the type of chemistry that could generate building blocks required for life. So I'll leave it there, and thank you very much for your time and for the invitation to come speak tonight. And I'm more than happy to answer any questions you might have. So thank you very much again.
Great. Thank you very much, Nick. That was a great talk. Very, very thought-provoking, I must say. I'm sure we have questions here. Who would like to start us off with questions? That there are no questions so far in the chat. Please feel free to write your questions in the chat. Maybe I could just start by, I mean, I guess you're looking for signatures that are indicative of microbial life. How is it possible that that there is some more morphological evidence of microbes in the rocks, and how would you go about looking for that?
Yeah, that's a great question, and something that I alluded to in the talk. The problem is that extraordinary claims require extraordinary evidence, and the science team and NASA are very aware of that. And that's the main driving factor behind eventually returning these samples to Earth, because I don't think we can be 100% sure that traces of ancient microbial life are recorded in these rocks unless we get them back to laboratories on Earth and study them in the way that we would ancient rocks from our own planet. I think the observations that the rover can make can help us select environments that we think stood the best chances of recording ancient life. But I think, short of seeing full-blown stromatolites, I think it's always going to be difficult to completely rule out some set of complex physical abiotic processes that could result in some morphological imprint in the rocks themselves.
I mean, I guess we've had rocks from the moon for a long time. Can you comment on kind of similarities and differences in moon rock and Mars rock samples?
Yeah, the Apollo era missions and sample return protocol is actually a great example and a great comparison to what's going on in Mars exploration right now. Our understanding of the moon was completely transformed when the first rocks came back from the Apollo 11 mission. There were a variety of hypotheses for how the moon formed, based mainly on a few lunar meteorites that we had from the Earth and a very limited sample set. But as soon as those rocks were returned to Earth, many of the alternative hypotheses were invalid. And the favorite working model that the moon was formed from a gigantic impact and basically reformed from Earth's debris after that major impact ended up being born. Now, given the circumstances under which the moon formed, we think that the moon probably never harbored enough water to really generate the types of environments that we think might have been favorable for life. But one really important set of lessons came from how NASA really handled the sample return missions. And the important thing was that NASA involved a good portion of the scientific community in analyzing those samples and did so in a relatively fair and transparent way. And I'm involved in a lot of the Mars sample return planning. So the group of scientists that get together and sit down and say, right, how should we plan analyzing the samples when they come back from Mars? What are some of the things we have to worry about? How can we do this in a process that is fair to the scientific community and is really driven by excellence first? And many of those lessons we learned from the Apollo experience. So it's quite valuable.
Yeah, thank you. I've got Cliff has got a question. Or any organic molecules found in it? And could I kind of tag on to that? I mean, of course, I'm kind of old enough to have got very excited by these prebiotic synthesis experiments that were done by people like Leslie Orgel and so on in the 1960s, which were really, really exciting. I mean, can we compare, what these prebiotic synthesis people found with what you might be finding in Mars? And also Cliff's question about organic molecules.
Yeah, so first off, organic molecules have been identified from Mars. The previous rover that was launched to Mars, the Mars Science Laboratory, which launched in 2012, had an instrument that allowed it to identify organic molecules. It was a pyrolysis instrument and a mass spectrometer, basically. And it identified organic molecules that are thought to be delivered from impacting meteorites mainly, and are the refractory type of organic molecules that are relatively common in those meteorites. I mean, that suggests that there's an influx of organic molecules that might have aided the transformation to more useful, biomolecules. But I don't think anything's been found that's similar to the type of building blocks that I alluded to at the very beginning of the talk. However, the story may well change once we get the rocks back and we can actually extract the leftover molecules in those rocks and really probe their structure in more detail.
Great. Well, Kirsten Hall he's got actually two questions in here, and one kind of picked up on the Leslie Orgel and Francis Crick thoughts about Panspermia hypothesis. I must confess I've not heard of that one myself, Kirsten. Life on Earth had been seeded by microbes from space.
Hey, Eric, I'll send you the paper. I was just looking at it the other day.
Sure.
They actually published it in a peer-reviewed paper. I'll send you the PDF of it.
All right. Okay. Thank you. That'd be interesting.
Yeah.
Nick,
Yeah.
Panspermia.
So my thoughts on panspermia is that this hypothesis originated in part from organic chemists that were so frustrated with understanding how to make the key biomolecules that they suggested that, well, maybe that they just came from somewhere else. And in fact, carrying that hypothesis even further, maybe life started somewhere else and basically seeded the Earth. In my mind, panspermia doesn't really address the problem of the origins of life. It just puts it somewhere else. And it makes it even harder because it doesn't really give us any environmental context. I think we can safely conclude that a lot of the ingredients required to generate the key biomolecules for life are present in some solar system materials. So meteorites, for example, amino acids have been found in meteorites and so on and so forth. But I think now we've made great progress in understanding some viable syntheses for how those molecules could be generated. And even more than that, how specific groups of molecules ended up being selected from a purely chemical process. So for example, certain types of amino acids were always thought that that biology actually selected for those amino acids for particular reasons. And it is true that they serve specific purposes. But a lot of the work that's come out of the labs of John Sutherland and others have shown that there are actually chemical reasons why those particular amino acids may have been generated in specific synthetic routes.
Thanks very much. Thank
Yeah.
you. Thanks for a great talk this evening. Absolutely fantastic. Thank you.
That was my pleasure.
Okay, Kirsten had another question there about microfossils in the ALH meteorite.
Yeah, sorry, I don't mean to hog the show tonight. I will shut up eventually.
Well, we deal with that one, and then we'll move on to Peter Henderson, I think. Did you?
They're great questions. Yeah, unfortunately I can't actually see the chat. Perhaps if I stop sharing, I could see the chat, and then. Oh, yes. Could you possibly update us on whether any definite conclusions have been reached about the possible presence of microfossils in the ALH meteorite that made the front pages of the newspapers back in 1996? Yeah, so the ALH purported microfossils were called microfossils on the basis of morphological evidence, and then on the basis of mineralogical evidence. So single domain crystals of magnetite were actually found associated with some of the carbonate minerals. And the first thought of scientists when they found those nanocrystalline magnetite domains was that this looks a lot like the type of magnetite that magnetotactic bacteria make on Earth today. So there's actually a class of bacteria that run their metabolism using iron, and they actually generate tiny crystals of magnetite to use as a sort of orienting device, or basically a compass. It was since discovered that just the thermal and physical processes associated with ejection of that meteorite was likely to have transformed the carbonates and to have generated the magnetite through thermal breakdown of the iron carbonate. And that hypothesis was shown experimentally and theoretically, and it was so convincing that it really provided a very simple abiotic explanation for some of the key pieces of evidence that were really forming the basis of that claim. And without those, most scientists now think that that was a really important cautionary tale in extraordinary claims require extraordinary evidence. I mean, it seemed too good to be true, and in the end, it's just very unfortunate that the entire set of observations could be explained with physical processes associated with the impact. So yeah, I think it's more of a cautionary tale than anything else at this point.
Thanks for clearing that up. That's great. Thank you.
No worries. Yeah.
Thanks. I mean, how about Peter Henderson's? Yeah, I should actually say that we've actually got quite a few biochemists in the audience tonight. And Peter Henderson is a biochemist too. How about getting these samples back to Earth? What about the mechanics of doing that?
Yeah,
is there a time when you've got to get them back, otherwise they get covered in dust or something?
Yeah, that's an excellent question. And there are a whole series of complicated questions related to preserving the integrity of the samples and making sure they aren't altered too much in the sort of 10 or so years from the time that Perseverance drills them out of the ground to the time that they enter the Earth's atmosphere. The plan now is that Mars sample return is a joint effort between NASA and the European Space Agency. And the Perseverance Rover mission is the first mission of 3 that will eventually return the samples to Earth. The second mission is called the Fetch Rover mission, and that's going to be a stripped down, relatively inexpensive rover that will land on the surface and go through and physically collect the cores that are left behind, the sample tubes that are left behind by the Perseverance Rover, and load them into something called the well, it's mainly just called the sample capsule, but it's this incredibly wonderfully engineered device that once the samples are sealed inside, ensures a minimum temperature fluctuation inside, and provides shock resistance to the cores themselves. So it's all cushioned and spring loaded. So the shock of blasting them out into Mars orbit and then re-entering Earth's orbit doesn't destroy the cores too much. So the Fetch rover will load that into the sample, the orbiting sample container. The sample container will be launched into orbit and will orbit Mars. And then the third mission that will sort of cap off the Mars sample return is an orbiter mission, and all its job is to rendezvous with the sample container, collect it, slingshot around Mars, and come back to the Earth and drop it into Earth's orbit. And the idea is that the sample capsule is going to land in a military test range in Utah in the United States. The engineers have ensured a minimum temperature window and a minimum shock window associated with that, but there are a whole host of other complications with merely drilling the rocks out of the ground. For example, there's a specific temperature and relative humidity range in the Martian surface, and even cracking those capsules open in the Earth's atmosphere could, of course, initiate a series of events that could irreversibly alter a lot of the minerals that are present in those rocks. So that needs to be done under controlled circumstances. And in fact, I was part of a group of scientists that really drilled into the details of this process and sort of recommended exactly how this should be done in order to make sure that the enormous investment into these missions isn't wasted by a relatively careless protocol.
Thanks. So Peter Reynolds has, I'll just read his question out. In the period where earliest life formed on Earth, as I understand, astronomers suggest the planet was subject to the late heavy bombardment, which in Earth's case potentially formed the moon. I think you might have mentioned that earlier, Nick. So was Mars subject to heavy bombardment, and could this have any impact on the formation of life though?
That's an excellent question. And so the answer to that question in short is yes. So as far as we can tell, the inner rocky planets of our solar system and the moon were all subject to relatively high flux of impactors early in their history, so about 3.5 to 4 billion years ago. The statistics of the size and number of those impactors has been measured on the moon and on Mercury and on Mars. And there's a very tight relationship associated with that allows you to date things as a function of time. But the big thing that probably influenced the origin of life were the very large impactors, impactors that were potentially so large that they could have, boiled off any oceans or lakes, changed the temperature of the atmosphere, and those impactors definitely happened on the Earth. They definitely happened on Mars. We can see the leftover craters. I mean, some of the craters are enormous. They're about 1,000 miles across or so. And so the consequences of those impacts are so dire that we think that any process that would have started life has to have occurred after the last big planet sterilizing impact, as it were. And so in a way, impactors are almost when the clock starts for the origin of life, or at least impactors below a certain point, if that sort of answers the question.
Just a couple, I heard on the radio this morning that I think the Oxford English Dictionary said that perseverance was was their word of the year, being the most searched.
It was the Cambridge Dictionary, but yes,
Cambridge
it was.
Dictionary.
All right.
Sorry.
So many people had been interested in this that they had actually managed to push that word up to the top. I was just reflecting. Thank you, everybody, for wonderful questions, including that quite simple question from Peter Henderson, which called forth a wonderful answer that was so interesting. I've always wondered so many of these things, including, I think, how on earth do these instruments manage to analyze the content of the rocks? I had no idea. So that fantastic for not for only a social scientist to hear that. And we're very interested to think of how 200 years ago, Nick, when it was the first season of the Phil and Lit in their new hall in Leeds, one of the lecturers was William Smith, who just made the first geological map of Britain. And you just think, what would he have asked you now?
Wow. Well, I have to say I'm humbled. I mean, this is a fantastic opportunity to come and speak at a learned society that really has a rich history, and it's been a real pleasure. So thank you ever so much for your invitation.
Well, thank you very much, Nick, and on behalf of all of us for a wonderful talk. Plenty of excellent comments about that in the chat. I think we've all appreciated this, and thank you again. Now, Rachel,
it's my pleasure.
Yeah. Rachel, do you have anything just in closing?
Well, yes, I'd just like to reiterate thanks from us all. And, part of the point of having a society with this wide brief of philosophy, as in natural philosophy, obviously that back in those days, and literature, and not getting at each other on politics and religion, but actually putting out there all sorts of intriguing and challenging ideas to be examined amongst people from very different backgrounds. And I think, this is absolutely in that tradition. So thank you so much for a really excellent session. Great. Thank you.
It was my pleasure. Thank you again.
Yeah. Okay, well, I think this is our last talk of 2021, and a fitting climax, I would say. I wish everybody a very happy Christmas and a good New Year, and we will see you all back in January. Rachel, do you want to say anything about our talk in January?
Yes, well, we're having a talk on the 20th of January from the relatively new Vice-Chancellor of the University, Simon Buitendijk. And then in February, we have Professor Westland carrying on our occasional series on colour. So he's going to ask provocatively, does colour exist? He's a physicist. And then in March, we have our new, relatively new, head of museums and galleries in Leeds, who's going to expound on the ideas about how the service is going to evolve under his direction. And then we've got a series shaping up for the rest of the year, and there'll be more news very soon.
Great. Thanks very much, Rachel, and thank you to you all. All the best, and take care.